Bispecific and multispecific antibodies are antibodies that can bind to 2 or more different targets. There are many different formats, such as the 3-chain, 4 chains, antibody fusion, and single-chain variety. Traditional monoclonal antibodies only need 2 genes to be expressed. They're the heavy and light chains. But bispecific and multispecific often require 3 or more distinct genes to be expressed, so there's often a challenge to express them with high titers. Each gene requires its own selection markers, so multi-genes require multiple selection markers. This can be challenging as well for bispecific and multispecific antibody, especially in the cell line development stage. When it comes to expression, the correct ratio of expression for each gene is very important in order to obtain the best quality products. It is critical that the expression of each gene can be tuned to that perfect ratio. Now, analytical testing is another area that can be challenging for antibodies modality. Verification of the ratio of the different chains is needed, and whether the bispecific or multispecific antibody bind to the specific targets as designed also need to be validated. Immunogenicity can be an issue for designing antibodies, but for bispecific and multispecific, there are so many different formats, pairings, structures to choose from. It is likely to choose and favor the design that is with the lowest immunogenicity. Finally, scale-up production can be an issue for some of the design, so I think innovators would need to consider all of those as they develop their bispecific and multispecific antibodies.